Welcome to Let's Chat About, the free monthly webinar series hosted by Sophia Sees Hope. We've developed this series with those living with Leber congenital amaurosis and other inherited retinal diseases in mind, but it's open to anyone who's interested in what's happening in our communities. My name is Alyssa Bass, and I'm the Director of Marketing and Communications for Sophia Sees Hope. Before we get started, I want to take a moment to thank our sponsors for this series: AGTC, Dominion Energy, Janssen, Mira GTX, ProQR, and Spark Therapeutics. We could not provide programs such as these without their support. A few housekeeping items. The session will last about 45 minutes. We have turned off the chat function during the webinar, but you can submit questions through the Q&A function. We did collect quite a few questions in advance from the registrations, and we will take those first. As time allows, we will take questions that come in live. We are recording this session, and after the webinar, we will post the recording to our YouTube channel and send all of the participants a link. It could take a few days, so we appreciate your patience. It's my pleasure today to welcome Emily Place, licensed genetic counselor and research study coordinator at Mass Eye and Ears Ocular Genomics Institute. Emily graduated from the University of St. Thomas with a Bachelor of Arts degree in biology. She went on to earn her master's degree in human genetics from Sarah Lawrence College. Prior to working at the Ocular Genomics Institute, she worked as a pediatric genetic counselor at the Children's Hospital of Philadelphia. Emily joined the OGI in 2011, where she provides risk assessment and genetic counseling services to families with inherited retinal dystrophies. She also serves as the study coordinator for the research study of molecular genetics of inherited retinal dystrophies. Welcome, Emily.
Thank you.
So we want to start out today by asking you to give us just a broad overview of genetics, which is kind of a huge question, but really with an eye towards how someone ends up with an inherited retinal disease.
Sure. Yeah, and thank you for the opportunity again to be here today and talk with you all about genetics and genetic testing. So yes, when we think about the topic of genetics is certainly very broad, but kind of the place I think we can start is discussing genes and kind of defining what that means when we're talking about genes, which are really the instructions and blueprint that tell our cells how to grow and function. And we have over 30,000 genes within our body. And we know of that over 200 have been identified to play a specific role in how the retinal cells should function properly. And all of this genetic information is really organized into our chromosomes, which is that, information that we can find in every cell of our body. And we inherit our genetic information from our parents. And so that's an important piece for us to keep in mind when we're thinking about the inherited conditions and specifically inherited retinal conditions. And so these inherited retinal conditions can arise because of a genetic variation or change within a gene that we know is important for retinal function. And what we really mean there is that this genetic change is changing the genetic code in some sort of way that's causing that gene not to function properly within the retinal cells. And so we know that these inherited conditions can be inherited in multiple different ways, and that's how they can appear within families. And one of the more common inheritance patterns is for these conditions to rise as a result of a recessively inherited condition. And really what we mean there is that this condition is resulting because of inheriting a genetic variation in two copies of a gene. One of those copies that we've inherited from mom and one of those copies that we've inherited from dad. And in these recessive conditions, generally there may be no family history of anybody else in the family with any similar conditions. And that's because individuals can be carriers in that they may carry one copy of a variation and one copy of their gene, but they have another copy that can compensate. And so carriers can be asymptomatic and run through multiple generations and not even know that they're carriers. And it isn't until two carriers meet that there's even a chance for both of them to pass on the genetic change. Other inheritance patterns can be, these conditions can arise because of a genetic variation on the X chromosome, or something known as X-linked disease or sex-linked disease. And typically in those conditions, we can see males more affected than females. However, certainly that can be different based off of families. And there may be a family history, maternal family history of other individuals that may be affected, but that's not, again, not always the case. And lastly, we know the IRDs can also be inherited in a dominant fashion. So going back to our 2 copies of every gene, a dominant condition can arise because of having just one genetic variation and 1 copy of that gene. And then typically in those families, we will see multiple generations that can be affected, but again, not always the case. And so that's where, and we'll kind of get to this in the following discussion, where really genetic testing can be helpful to better clarify what is that underlying genetic explanation and how is this condition arising within the family, and thus better provide a more definitive diagnosis and risk assessment to other family members.
So I'm going off script here, but you made me think, understanding genetics has existed for centuries, but really knowing the full map of human genetics only came about completely in the 80s, right? Is that correct?
So certainly the sequencing of the human genome was done actually back in the 90s. And so our understanding of the genetic information and genes that can be associated with human disease continues to evolve. So a lot of work has been done to identify many of the genetic causes of inherited conditions in general, but there still certainly is a lot of work to be done to better understand the causes of inherited conditions and identify more genes, because we know not all of the genes have been identified to date.
So tell us about the role of genetic testing and a genetic counselor, the role that a genetic counselor plays in the life of a patient who has an inherited retinal disease.
Yeah, and so kind of going back to what I had said initially, so we know that over 200 different genetic genes have been identified to cause inherited retinal diseases. And I use that again, that term is kind of a larger category with LCA or Leber's congenital amaurosis being a type of inherited retinal disease. And really in general, though there's a few exceptions, we can't really tell or predict what the underlying genetic explanation can be for someone's retinal disease just based on an exam alone. And so that's where genetic testing is necessary in order to rule in or identify which one of these, again, 200 plus genes could be the underlying explanation, and thus more definitively rule in an inherited condition and provide a more definitive or specific genetic diagnosis. And counselors can play, are involved kind of in multiple ways in the process as a family or patient is working towards getting a genetic diagnosis. And it really can start with meeting with families when in kind of what we think about is the pre-test counseling portion, where obtaining that detailed family history, reviewing what the different inheritance patterns can be, and reviewing the options for genetic testing, what's available, what are going to be the benefits and limitations of doing genetic testing, what can we learn from genetic testing, what can't we learn from genetic testing, and what are the type of results that we can get back. And it's also exploring with patients and families, is this the right time for genetic testing? Is this something that the individual is ready to move forward with, or is it something that maybe should be discussed or pursued at a later time? When results come back, then once genetic testing results come back, a counselor can be involved in reviewing those results with the family, going over specifically what was found through the testing. Was a specific gene identified that we think is the underlying explanation? Or was the testing negative where no clear genetic explanation was found? And if that's the case, how does that play into the patient's care going forward? If a specific gene was identified, what does that mean in terms of risk assessment for other family members and helping coordinate our other family members should get genetic testing? And how does that information be helpful for thinking about genetic related treatments and therapies? As now that's more important within our field. How does this information inform about therapies going forward? And so what I've really found over the years, this is a really long-term relationship we have with families. So once testing is done or a genetic or gene is identified, we still have an ongoing relationship in terms of how does this information continue to impact on the family? Are there other family members that need to be tested? And how do we maintain our communications going forward about potential treatments and therapy options?
That's interesting that you say it's a long-term relationship, because as you were describing the process, it sounded to me like the genetic counselors are involved in the very beginning of the patient journey and then aren't needed anymore. So that's interesting that you actually have a long-term relationship.
Yeah, I think every clinic and counselor's experience will be a bit different, right? I have the, I think, true pleasure of being involved with within an IRD clinic where we've have long-term relationships with our families, and I get to see families back over the course of their visits with us. So it is a true privilege to be able to have create these longer relationships with families. Sometimes that may not be the case with the counselor that you speak with, that they're there for just the pre and post-test counseling, then they're working with your ophthalmic team to help provide inform your care going forward.
And just talk quickly about the mechanics of DNA testing and gene testing. You know, there's all of these commercial entities now where you can find out where all your ancestors came from and all of that. And that's not what we're talking about. So talk about the mechanics of this kind of genetic testing.
Yeah, right. A number of different types of genetic testing is available out there. What we're really talking about here when we're talking about testing for these inherited retinal conditions is diagnostic testing that's performed at a clinically certified lab that's analyzing a specific set of genes that have been identified to date to cause. We know when a variant occurs within that gene, it can result in a retinal condition, versus the direct-to-consumer testing, which is looking at variants in regards to risk assessment to more common complex genetic conditions.
And is it like you see on TV where you swab the inside of your cheek and put it in a test tube and mail it off, or what is it? Yeah,
so the sample types that can be, this testing can be done on does include the swab. So there are saliva-based testing where it's either you provide a saliva sample, or so-called spitting in a tube, or using a swab to brush the inside of your mouth, or it can be done through a blood sample. And we're really finding that blood samples or a saliva sample really perform at the same quality and quantity. So the saliva samples in this day and age, right, are facilitating testing for patients much easier, because that can certainly be coordinated at a distance through many telecounseling appointments.
Right. Over the course of your career, what have you seen in the area of inherited retinal diseases that encourages you, and what have you seen that discourages you?
Yeah, so this was a good question, because it got me thinking over kind of what's happened over the last, I guess, about 10 years, right? And so when I started my career within this space, the first clinical trials for RPE65 were just getting started. And so where we've come from, or starting at that point in terms of where we are today, we have a licensed gene therapy for RPE65. And we have so many more clinical trials that are starting to explore genetic-based therapies for many of the other genetic forms of disease. So I think that's really encouraging. Also that, gene therapies are and different types of genetic-based therapies are being explored, but also drug-based therapies and stem cell therapies. So I think it's important that different types of therapeutic interventions are being researched. And I think at some point we'll see a combination of these different types of interventions that will be most helpful for patients. The discouraging part, and I think this may echo a bit about what families and patients feel as well, is that it can sometimes feel slow, that we all wish things we had something right now, and that that science would be moving faster. And so I certainly do wish, I can feel that as well, working within the field. We just hope that that things would move more quickly.
So 2 questions came in through the registration process, and I grouped them together because they're a little bit different, but I feel like they'll resonate with a lot of people in our community. Because of the rarity of all of these diseases, we often hear from patients as an advocacy organization that they cannot find a doctor to help them. And so one question that came in was very specific: how do I get my daughter, who is 40 years old with LCA and had a kidney transplant, tested when her doctor does not help? And then the other question, which is really one that we hear on an almost daily basis, is what do you say to an ophthalmologist who doesn't always order a genetic test for a suspected IRD? And people who aren't lucky enough to live near a major metropolitan area like Philadelphia or Cincinnati or New York or Boston or San Diego, they really have a hard time finding somebody who can help them get started on this diagnostic journey.
Yeah, and I really appreciate these questions because it's, I certainly can hear them as well in terms of how do we better provide resources within the community to help facilitate genetic testing for our patients. And certainly thinking about one area to turn to is identifying genetic providers within your community, whether that's a genetic counselor or a medical geneticist, which is an MD. And those individuals can be found depending on your location and your hospital system in a medical genetics department or a clinical genetics department. And so both the National Society of Genetic Counselors, as well as the American College of Medical Genetics, do have search engines on their websites to put in your location and find genetic professionals within your region. And I would say that's one area to consider, finding the genetic professionals to help facilitate testing. The other is that, more and more these, I guess 2 pieces. One is that there are telegenetic counseling companies that are becoming available that offer telegenetic counseling services where you would meet with a genetic counselor, again, over the phone, over a WebEx, and have that session, and then they can work with you and a provider to help order the genetic testing and then be there when the results come back as well. And so that's another way to be able to connect with a genetics professional, again, who may not be in your area, but at least who's knowledgeable within inherited records.
Because I can't just get myself a genetic test, right? I need a doctor to.
That's right. So these tests do need to be ordered by a medical provider. So whether that's an ophthalmologist, a primary care provider, or a geneticist or genetic counselor.
Okay. And this one is very specific to a certain gene. With a recessive CEP290 variants without a second marker found in, I don't know how to pronounce this, XOME sequencing, can one still be diagnosed clinically with LCA? And I want you to answer this question, but this is so technical. And I think if you could just talk for a second about moms and dads getting all of this kind of medical jargon and talk and very specific biological information tossed at them by somebody who's examined their child who has low vision. It can become very overwhelming, I think, for just regular people who are trying to figure out what's going on with their kid.
Yeah, and maybe one piece, but before I kind of get into the specifics of that question, is kind of going to the question, or the second part of that, the other question you asked about the ophthalmologist maybe not being as comfortable with ordering a genetic test, which that's where I would advocate for, again, a genetic counselor, a medical geneticist that can be involved in the approach to ordering genetic testing. So I think it's hugely important if a ophthalmologist recognizes, I think genetic testing could be helpful here in providing a diagnosis. And that's where reaching out to colleagues to help facilitate that process, to have those difficult conversations and those technical conversations when results come back, to be able to take the time and go through, okay, this is the testing that was done. These are the results that were found. This is what we think in terms of the next steps of testing that might be needed. And kind of with these sponsored testing programs, there are genetic counselors associated with them that can provide that post-test counseling. So I would like to, it's a reminder to the ophthalmologists who may not be comfortable ordering the testing that you already have support built into some of these programs to turn to help with those results discussions with your patients. And so that kind of gets to this question about the CYP290 variants that were identified through testing, and it's the results we can get back is complicated in terms of the testing that's done, because a lot of this testing is quite broad and a lot of different genetic variations can be returned. And how do we make sense of that? And did we find an answer that we think is the underlying explanation? So I guess to start with, we think about LCA as a clinical diagnosis, and that's made because of observing certain symptoms on a clinical evaluation. And the genetic test is really there to help, again, rule in what the specific underlying gene may be. But we know that for about 30% of patients, we don't find a clear genetic explanation when we do comprehensive testing or that exome sequencing that the question was asked. And that may be that the testing doesn't identify a specific genetic explanation, but we still think there's probably is a genetic explanation for why. Or in the case of this, sounds like this family, is that testing identified one single genetic variation within the CYP290 gene, but it didn't find a second. And we know CYP290 is associated with recessive forms of disease. So in order to confirm a diagnosis of CYP290, we need to identify two mutations. And so identifying one, I think from my perspective, is this may be a hint. This is the underlying gene, and this is showing the limitations of our technology right now, that there are probably further testing of the CYP290 gene that could be done to better interrogate, could we find the second mutation and thus confirm the underlying genetic diagnosis for a patient. But I think this kind of highlights some of the limitations of testing, is that we're not always able to identify a clear genetic explanation, and that there is more testing that could potentially be done, whether that's diagnostically or potentially through research-based testing.
I guess too, it's a good indicator of how just because there's a treatment for a specific CYP290 variation doesn't mean that there will be a treatment for every CYP290 variation.
That's right. So when we're looking at gene-related therapies, we need to think about, is that therapy related to, in general, for the gene, and it doesn't matter what the specific mutation is, or is that treatment or intervention very specific to not only the gene, but the specific mutation or the region in which that mutation falls.
And we see that currently in the research and in the trials.
We do, yes, yeah. And so I think that's another important piece with the follow-up counseling discussions, right? Is identifying, well, how does this information inform about therapies right now, and could it be informative about therapies going forward?
So we have some great questions that have come in through the Q&A. I'm going to ask you this one first. I think it's such a good question, and we hear this one a lot too. I'd love to know the difference between a negative and an inconclusive result. Yeah, so
and I think how a lab kind of reports that out may be a little bit different. A negative result can mean that there were no genetic variations that were identified through testing that are the likely underlying genetic explanation. Inconclusive?
An inconclusive versus a negative.
An inconclusive result may suggest that there were variants that were identified within a gene or two genes that the lab just doesn't have enough data currently to be able to classify those variants as disease causing but they also can't rule them out as not causing disease. And sometimes in the inconclusive results, additional testing of other family members may be helpful in resolving what the significance of those variants, or that it's going to take some time, more genetic knowledge is needed in order to better classify those variants.
So I could see in cases like that where someone would really need a genetic counselor, because I would imagine if I were a parent and I were trying to find out a diagnosis for my child, an inconclusive result would put me into counseling.
Yeah, I think that's where working with a counselor to talk through what was actually found for this testing. And again, what were the recommendations in terms of next steps? Was the lab willing to test other family members because they think one of these variants may be the explanation, and that is one piece of evidence that's going to be important for reclassifying the variants versus is it just needing more time? And that, I think, speaks to the continued relationship with genetics going forward, is that if a negative result or inconclusive result is received, it may be helpful to check it back in with your genetics professional every year, every couple years, to say, has anything changed? Is there new testing available? What do we know about these variants that we didn't know 2 years ago? And has their classification changed, and does that impact my result?
Are they refining and improving genetic testing all the time? Is it like everything else where they just keep working on it?
They do. So certainly as new genes are identified or technology changes, more comprehensive testing may be available. So especially thinking about for families that may have had a negative result, there may be additional testing in the future that would be appropriate as more genetic knowledge is known and testing changes.
So this is a long one. While sequencing genome in a mosaic disease, they say one cell does not represent all cells. How important is it to sequence different types of cells? Why in some mosaic patients, somatic cells repair themselves spontaneously without an external intervention? Have you come across any such cases? And if you could help me out and explain to me what a mosaic disease is, then I think that would be helpful in answering this.
Sure. So when we think about mosaic disease, can mean that a variation is not found in all cell types. So kind of going back to the fact that I said that our genetic information is found within every cell of our body. Well, sometimes variations can arise that may only be found in certain specific cell types. And so, when we do genetic testing for these inherited retinal conditions, we're taking a saliva sample, as I said, or a blood sample, thinking that cell type is going to be representative of the cell type that's found within the retina. And again, we're thinking of these as changes someone inherited, so we would expect them to be present within all cell types. And so I guess there's one thing to potentially keep in mind right there, is that maybe we're not doing genetic testing on a blood sample or saliva sample may not be totally representative of what the cell types or what's going on within the retinal cells. So maybe there are changes that we're missing there.
Okay. This is a good one. I'm curious if there's a sense of how often IRDs are misdiagnosed today versus say 5 or 10 years ago. Is there a chance for misdiagnosis with genetic testing?
Yeah, I think that's a great question. I don't know I don't have any specific numbers there. I think less often patients are misdiagnosed now than they were maybe 5 to 10 years ago, because knowledge is certainly expanded and awareness of these inherited conditions have. But I think one thing we've learned from genetic testing is that we can't always, it kind of going back to that clinical question or the clinical observation, is that we sometimes get our clinical diagnosis not as correct as we thought it would be, because we're using the symptoms to help create that clinical diagnosis. And ultimately the gene is going to be the most helpful information in for better defining the condition.
How do patients and their families feel about registries? Do you feel that that they know about them and they understand what they're for? And I think as part of that, if you could speak to what you think about registries, patient registries, where people put their information in with their genetic diagnosis, how important you think it is for people to participate in those as well.
Yeah, I think in my experience, in my clinical experience with the patients that we've seen, are very supportive of registries, and that many of our patients appreciate the opportunity to even be involved in a registry and feeling like they're contributing to the science, and have the opportunity to stay informed about research opportunities going forward. And I would agree as well. I'm supportive, and that's where I talk about it with patients. Is this is an option. This is something that's available to you. If you want to learn more, here's where you can get more information about the registry. And if you want to participate, please let us know what we can do to help facilitate your registration process.
That sort of makes me wonder, what kind of relationship, if any, do genetic counselors have with researchers and with companies that run clinical trials?
I think you can kind of see those relationships in all different forms throughout the kind of community, where genetic counselors can be more intimately involved in clinical trials that are going on, or where kind of in my setting, where I work more closely with the lab scientists in terms of doing disease gene identification and really trying to help identify new genetic causes of disease, or where counselors may be working with those companies that are developing those treatments and interventions and helping to work with the patient advocacy groups to identify patients and get the word out about potential opportunities.
Can you speak a little more about the progress of RPGRIP1 research?
Yep, RPGRIP1. So there is research going forward to try to develop gene-related therapy for the RPGRIP1 gene, one of which that had started in the academic setting, actually at Mass Eye and Ear, and now a company is working to take those trials forward in creating a gene therapy there.
How challenging is it for patients who do have a genetic diagnosis to find out what's going on in the fields of research and treatment development? We get those kinds of questions all the time as an advocacy organization, but it seems challenging.
I agree there. I think it is a challenge. How do we, as a community, stay up to date with what's going on in the research kind of realm, and what's the status in terms of how different gene-related research is going on. And so I guess I don't have an answer to the question, but I think there's a huge need for creating some sort of dashboard or database or a way to be able to kind of keep track on what's going on for each specific gene.
This question says, my daughter's genetic test showed she had a de novo autosomal dominant CRX mutation. Is there any reason to have the parents get a genetic test?
So when we use the term de novo as a new genetic variation, so meaning that that a variation arose because it doesn't appear to be inherited from either parent. And so I would say that the importance for doing parental testing is to confirm that piece, and that can be helpful for better understanding risk assessment potentially to other family members.
So I just want to make sure I understand this. Does that mean that the child has this genetic variation not because of inheriting it from the parents? Like it's just a random variation that happens in nature?
Yeah, so we know that so we all have a handful of genetic variation our parents don't have. And sometimes that variation can fall within a gene that can cause a condition, or specifically a gene that can cause dominant disease. And we know for the inherited retinal disease, that happens about 1% of the time. So while it still falls in the inherited category, it wasn't specifically a variation that was inherited from a parent, but it was a new genetic variation that arose at the time of conception.
Is that a more challenging diagnosis for someone to have than someone who has your regular run-of-the-mill inherited variation?
I would say from the post-test counseling side that these are results that we know can happen, but they certainly can be a surprise because there's no family history of anyone else in the family affected. It can increase the patient's risk for passing this variation on to future offspring. And so I think can be some challenges around explaining the results in terms of how it arose.
Okay. This question, it says, and so I guess this is going to be something that you can explain as well. Do you recommend genetic testing by disease, or do you prescribe full genome, full panel to all patients? So I didn't realize that you could order very specific genetic testing, or you can order full bore.
Yeah, so there's different types of genetic testing that can be performed. I would say really my approach to testing is to do full IRD panel testing for any patient with a diagnosis of any type of inherited retinal disease. What we found, if you kind of look through multiple different commercial entities that have available, they have a panel that's for just RP, or just cone rod dystrophy, or just macular disease. And I've learned from experiences that we're not that good about picking the underlying genetic explanation. And so that's where full panel testing is actually really important, because we may miss the gene if we did only looked at a specific number of genes. I would say I would do full panel testing of inherited retinal disease genes before I would go off and do whole exome or whole genome sequencing. At this point, I'm not sure the clinical, for inherited retinal disease, the clinical utility of doing those even broader types of tests.
Okay. And this I like because it says, what would you say to an ophthalmologist with a patient who had an inconclusive test a few years back? Should they re-engage that patient to get them retested? So I love this because it sort of raises the idea of genetic counseling of ophthalmologists and not just patients and patient families. So talk about that a little bit.
Yeah, I think it's a good opportunity to re-engage the provider, to say is to re-engage the provider to re-engage the family. And at that point, it can be, well, maybe we should go back to the testing lab that did the test a few years ago and say, has something changed on their end? Has more data helped redefine the variants that were identified? Do we know something different now than we knew a few years ago? Or is this really a point where a few years ago testing was limited, only a smaller number of genes was tested, that this patient could really benefit from more comprehensive testing?
So really it's important to have a back and forth relationship. It shouldn't just fall on the patient and the patient family all the time to be circling back and saying, how about now? How about now? How about now? Right.
It's definitely a two-way relationship, right? So continuing to stay engaged with your providers, but your provider also thinking about being on top of the different testing options that are available, and being aware if there's something more comprehensive or more appropriate that would be better at that point in time.
All right, well, we've run through all of the questions that were submitted in advance and also through the live webinar. Terrific questions. Thank you so much to everybody. Emily, if you had to leave us with a piece of inspiration, what would it be?
Oh, I think if so much has changed in terms of our access to genetic testing for inherited diseases over the last several years. And hopefully, although it's more available to patients than it ever has been in the past. And so I would say this is really a time to work towards getting a genetic diagnosis or getting genetic testing if you haven't had it, or re-engaging with your providers if you've had it years ago, and consider doing it at this point in time. There are the resources out there and available. So there are plenty of individuals like myself that are available to help facilitate testing or get you in touch with the right counselor or medical geneticist to help facilitate testing for you.
All right. Emily, thank you so much for all that you do and for your longtime support of Sophia Sees Hope. We greatly appreciate all the work that you do.
Thank you.
All right. Thanks, everyone. We will follow up with an email with the link to the video and some resources.